In this lecture, we will focus on conservation score. In the previous class, we discussed about the multiple sequence alignment. Right? If you align different sequences, right, we will get alignment, and how to utilize this alignment to extract different features. How far we see that a particular residue is highly conserved, right, in any positions, right. So, what did we discuss in the last class? We discussed a blast. What is a blast? Basic local alignment search tool, right. So, what is the software is called blast, right. You can, what are the various features of blast? What are the applications of blast?
Either through database we can align, or two sequence we can align.
If you have a query sequence, you can identify the sequences which are similar to your query sequence. If you have two sequences, you can see the identity or similarity between the sequences. Then you can use blast to identify any specific motifs or any specific patterns, right, inherently present in any sequence or in functionally important motifs, right. So, they can use either DNA sequence or the protein sequence, right, for any sequence alignment. Then how to evaluate whether two sequences are similar or not? What are the various scores available to evaluate the alignment?
Alignment score.
Alignment score depending upon the total, can do with the different aspects, right. One is you can with the matching pairs, right. So, then the second one you can do with the similarity, then we can do with the identity, then the query coverage, then p value or the e value, right. You can see the probability of having that alignment with the specific threshold score, right, or the what is the expected value. Then we discussed about the multiple sequence alignment, right. How many sequences are required for multiple sequence alignment?
More than two.
More than two. Minimum you need 3, right. 3 or more sequences if you have, right. What is the principle used in multiple sequence alignment? How they do the alignment?
One by one.
Right. They have the pairwise alignment. Then see the similar sequences put together, right. And then they can align these disintegrated sequences. Finally, they make the complete alignment. Right. What is the program which can give the multiple sequence alignment? Cluster. Also, we discussed few more software. What are the other online resources? Mapped, muscle, promos, right. This can do that. Then we discussed about SciBlast. What is SciBlast? Position specific, right, rated the blast. So, here you can make the position specific scoring matrices forward sequence, right, by aligning with the different other sequences, right. They can use different iterations, right, with any threshold value. So, finally, you can get the scoring matrix. You can also call as profiles. So, now if you have a multiple sequence alignment, what are the applications? What can we infer from multiple sequence alignment? There are several applications, right. So, today we will discuss about conservation. So, what do you think about conservation, right? If you think about conservation, see, if you have set of sequences, some positions are occupied by same residue or similar residues, right. In this case, you can say that position, right, maintains a particular amino acid residue, right, either in protein sequences or nucleic acid sequences. So, and this sequence also depends upon the evolutionary rate, right, how frequent a residue is mutated to different residues. Likewise, when we discussed about the PAM matrix, we have the alignment, several residues are the same and some residues change, right, during evolution. So, likewise, the score also depends on how far a specific residue is mutated, right, in the homologous sequences. So, whether this rate of evolution is same or different, that depends upon the amino acid changes, right. Some cases it remains the same. In the PAM matrix also we discussed about the different amino acids, right. For example, cysteine or tryptophan, so you get the high score, right, and some positions which evolve slowly to the different residues. Some cases they maintain to be constant and some positions they change from organism to organism. So, the positions which are the same, right, these are called the conserved. So, the ones which evolve rapidly, that changes in different organisms, they are called variable. So, if you have multiple sequence alignment, you can look at the 3 positions. In some positions you can see that residues they maintain at the same location, they are called conserved, and some cases different organisms they change the residues, they are called variable. Then how to get the conservation score, right? For example, if you have an amino acid sequence, you like to know which residues are conserved and which residues are variable. How to do that? First we have to get the sequences, right? For the query sequence you will get the similar homologous sequences. Where shall we get the homologous sequences? BLAST. So you can, if you go to BLAST and you get the homologous sequences and you get the sequences, right, and do the multiple sequence alignment and then you get the score. So you have to compare the sequence of similar proteins, right, deposited in the UniProt or SwissProt database and which are homologous to this your own sequence, right. Then you can compare the sequences and you will see where you have the variable residues and where you have the conserved residues. So now if you give the multiple sequence alignment, for example, if you see position number 60. So if you look at the position number 60, so this position is conserved or variable? It is conserved, because if you see the 10 different sequences, okay, this is the UniProt ID, okay, here this is the sequence, okay, just for example I kept from 1 to 60. We take the 60th position, you can see all the residues, right, these positions, right, are accommodated with the residue glycine. If you look at this position, right, 1, 2, 3, 4, 5, 6, 7, 8, 9. So this position is variable or conserved? It is variable and we can also see conserved. Okay, if you see this position, so what are the preference of amino acid residues at this position? T, A, G, S, right, T, A, G, S. But if you look into the all the 10 sequences, most of them have the residue 309. Now if you have this alignment, how to give numbers, how to get score? So which one is highly conserved, which one is highly variable, right? Because some cases if you say the position number 6, here also you see the position is accommodated with the alanine, D, E. So here also 3 residues, here also 4 residues, right? Which one is variable, which one is conserved, how far the difference between these two positions. So we need to give a score. If you numerically get some numbers, then you can compare, okay, this position A is more conserved than position number B. So how to do that? This involves 2 different steps. First step, we need to get the amino acid frequencies. Each position, how far each amino acid residue prefers at a particular position, and then the second part we convert these frequencies into scores. So there are various ways to get the frequencies as well as to get the score. If you see the frequencies, okay, I listed up 3 different ways to get the frequencies. One is unweighted amino acid frequencies. Here we do not give any weightage, all amino acids are treated samely, all sequences are treated samely. So we do not give any weightage to any sequence or any positions or any amino acids. And the second one, we give weightage to some amino acids or to some sequences. For example, if you have a particular sequence, right, we know that we need to maintain the residue positions, then we can give some preferences, right, we have to maintain that particular sequence. Likewise, like some positions, for example, some cysteine is very highly conserved, we know that this forms disulfide bridge. So if you want to keep that, we will give weightage to that particular residue, right. So if it is cysteine, then we give more weightage, otherwise you give less weightage, you can do that. Then third one, you can compare with the random choice. You have your aligned sequences, then if you shuffle randomly you will get the sequences and how far it will match your aligned sequences as well as the random sequences, right. Then in this case you can say, okay, this is your aligned sequence which are really significant and you can calculate the frequency of that particular position. So if you get this preference, then we can convert this into score, because we need numerical values, right. So either we can use entropy based method, right, considering the probability of residue at a particular position, take the probability multiplied with logarithmic of probability, you can get the score. And the second one, you can do the variance based measure, right, how far these positions vary with different positions. For example, alanine at position number 3, how this differs from alanine in position number 10, that how many alanines in position number 3, how many alanines in position number 10, and totally how many alanines a sequence, compare with all these positions and the residues, we can measure a score. Then you can also use sum of pair score. For example, if you sequence A and you can the second sequence B, so you have two different amino acids. So you can use a matrix to see how far they are consistent, how far they are aligned, right. Which matrix usually we use? Pi matrix or Blossom matrix. You can see and you put proper weightage and see whether which one is highly conserved, which position is highly conserved. So, I will explain the steps one by one. Okay. First we go with the unweighted amino acid frequencies. Here we do not give any weightage. In this case U means unweighted. So, frequency of residue A at the position I, this is given as number of sequences having the residue alanine at position number I normalized with the total number of aligned sequences. So, this I varies from 1 to 20, right. This is equal to 1 to 20 for the 20 different amino acid residues. So, if you see this alignment, what is the frequency of occurrence of this amino acid residues at position number 9? How many times T?
6 times.
6 times T, 6 T's,
7 times.
7 times T, 7 times A,
1 time.
1 time G,
1 time.
1 time S,
1 time.
1 time, right. If you have another position, for example alanine 7 times and any of the other residues 1 1 1, so you will get a similar score or different score? Similar score, because here we do not use any weight to any different amino acids, we give equal weightage to all amino acids. Even in this case, if it is instead of threonine 7, if it is valine 7 times and so alanine 1, tryptophan 1, right. So, then we will get the same score, but if you look into this alignment, sometimes if you have the threonine, it will not match with the hydrophobic residues, but even in this case you will get the same score, because we do not give weightage to any amino acid residues. So, here f of I is the each residue I, right, any residue for A in the any position I normalized with the total number of sequences at a particular position, fine. So, normally we use this for a specific group of sequences which is highly homologous, any specific group, then we expect similar residues at similar positions. So, in this case we use unweighted amino acid frequencies for any specific group of sequences. So, the second one, this is weighted amino acid frequencies. Here we give weightage, if we give weightage to any specific sequence or we weightage to any given residues, right, to give preference. Here we introduce this term, right, delta AKI. So, this is equal to 1 if A in the sequence K at position I, if you decide position, right, any position I in the sequence K, so amino acid A is in the sequence, right, and then you give the weightage for that particular sequence. Otherwise, this is equal to 0. Wk is the weightage of a sequence K, right, and delta AKI equal to 1 if the amino acid A is in the sequence K at position I. If it is not, then you can put 0 otherwise. So, the K equal to 1 to number of sequences. So, here why we need to do this weightage position control, also we give some preference to some specific amino acid residues. If threonine 6 times and 4 hydrophobic residues, this different from threonine 6 times and serine 4 times. So, similar type of residues you can have more score. So, in this case we give weightage. Then also you can give some sequences more weightage. For example, if you have several sequences, right, and some of them are highly related and some of them are distant related. If you want to include the distant related sequences, for example divergent sequences, if you want to include this in the score, then you have to give weightage to the sequence. Otherwise, if 9 sequences are homologous and one is distant related ones, so the score will be always high, because based on the 9. So, if you want to include the preference of this divergent sequence, then we need to give a weight. So, in that case you can include the information from these divergent sequences. Then the third one, we can also use this frequency based on dependent counts, how far you can get these counts in random distribution. So, independent counts they put IC, the same the frequency of occurrence of any residue A at position number I, which is given as number of residues of A at position number I normalized to the total number of residues at any position. So, if all the 20 residues are at the random distribution, then you can assume that f equal to 20 into 1 minus 0.95 into n. if n equal to 1, what is the value? 1 minus 0.95, this is equal to 0.05, 0.05 into 20, that is equal to 1, right. So, now you can see you can assume that we can fit this function f in this equation such a way that if it is totally random, completely 5%, so you can, if you do this equation, you can get the 1 equal to 1, f equal to 1 is what expected, that is fine. So, from this one you can calculate the effective of this n, you have the actual values and you have the random values and you can fit with some function, right. In this case you can fit with this n as n effective. So, use this equation, then if you change this equation to get the n effective, right. So, you move this 20 here, that means f by 20 equal to 1 minus 0.95 into n power n. In this case 1 minus f by 20, this equal to 0.95 power n. Then take the logarithm ln, right, ln, then you can move this n here, that is n into ln of 0.95. From this you can get this n effective, right, this equal to ln of 1 minus f by 20 divided by ln of 0.95. So, you can get this number, right. So, for any f, take amino acid A in position I in a single sequence, this equal to 1 if you take that. Then if you have more sequences, for example 20 sequences, if you have one more sequence, there are two possibilities. One possibility is that could be the same amino acid, right. For example, if you have a sequence, right, A I T S T A, right, this is a concept here, right. Then we add new sequence, there are two possibilities. One possibility is it could be A, then this case NAIC of I is equal to 1 because it is identical, even if you add more, there is no difference because average is the same. And second possibility is other than A, whatever it is, because if it is other than A, it is not identical. If it is not A, then NIC of I equal to NA of I, because it depends upon the amino acid. So, now the question is, if you add one sequence, what is the probability of this function f, right, whether this will fit with this equation or not. So, in this case imagine the equation with two conditions, one if n equal to 1 or n equal to n plus 1. If both the cases, if you are able to fit in this equation, then you can say that you can use this to get the independent counts. So, if n equal to 1, then it will fit with this equation, n equal to 1, f equal to 1, that we discussed earlier. So, that is true. Then assume n equal to n, where f of n I is a probability of I, right, different amino acids to occur the position. So, now you can see this, for if n equal to n, you will get this equation f of n I comma I, that is equal to 20 into 1 minus 0.95 into n, right, that is fine because we assumed it, that is fine. Now, if n equal to n plus 1, okay, then you add one more sequence, then there are two possibilities, one is it is I by 20 adding the same one, or we can take the I plus 1 with the probability of 20 minus I by 20, there are 20 possibilities, not the same one. So, I got to subtract, so 20 minus I divided by 20, we have 20 different possibilities. Then if you add this in this equation, f equal to I into I by 20, right, and I plus 1 into this 20 minus I by 20 into function of n comma I. So, if you simplify this equation, you will get this 1 plus 0.95 into I with the function of n comma I, and this equal to 20 into 1 minus 0.95 into n plus 1, because we derive these numbers, and if this is equal to this 1, if you divide by 20, right, 0.05, then finally if you take this out 20 out, then you will get 20 into 1 minus 0.95 into n plus 1. So, if you look these two equations, if n equal to n or n equal to 1, it obeys this equation, and if it is n plus 1, the same, instead of n we use n plus 1. So, in this, if you prove this one, then we can use this particular equation to see the actual and the random distribution and to get the independent counts.